All right, guys. Today we are going to talk about non-Mendelian inheritance. So the Punnett squares we've done so far are when there is a normal situation of dominant and recessive. And in that non-Mendelian inheritance, sometimes things don't work quite that way. So we're looking at SB3B, where we are going to analyze how biological traits are passed on to successive generations. And we need to be able to use mathematical models like our Punnett squares to predict and explain patterns of inheritance. And our goal for today is to explain the difference in patterns of inheritance that don't follow Mendel's laws. So we know that Mendelian genetics shows us that many traits are dominant over others, and that for a recessive trait to show up in the phenotype, both alleles have to be recessive. However, now we know that there are some exceptions to that. There are some alleles that are neither dominant or recessive. Some traits are controlled by more than one gene, and the majority of genes have more than two alleles, not in your own body. We only have two copies in our body, but in the gene pool as a whole. So let's see what the amoeba sisters have to say.
What do snapdragons, human height, and speckled chickens have in common? Well, they are easily identified as non-Mendelian traits. By non-Mendelian, we mean that genetically, they are rule breakers. They don't follow the regular Mendelian rule that having a dominant allele means the dominant trait will show. Remember in our guinea pig video, having a dominant allele represented by a capital H meant that the guinea pig would have hair? Only if there was no dominant present, a genotype of little h, would there be a hairless guinea pig. Well, that's a Mendelian trait. But what we're going to cover now is when these traits are non-Mendelian and they don't follow these basic rules. So let's first start by snapdragons. We confess that when we heard this word, we thought they were some really amazing kind of creature. Well, they are amazing, but they're flowers, so I don't know, that's not exactly what we envisioned. Well, in snapdragon genetics, there can be three phenotypes: red, white, or something in between, pink. It's called incomplete dominance. In incomplete dominance, the dominant allele is not completely expressed when the recessive allele is around. There isn't really a clear dominant allele. If you cross a red flower, and we're going to write it here big R, and a white flower, little r, you are going to get offspring that are big R, little r. But unlike a Mendelian trait, if this is incomplete dominance, that big R allele is not completely expressed when the little r is around. So big R, little r in this case is pink. If you cross two pink flowers, big R, little r, like shown in this Punnett square here, you can get offspring that are red, white, or pink.
All right, guys. So incomplete dominance is the first type of non-Mendelian inheritance. And with incomplete dominance, we ultimately have offspring that, if the heterozygote is the genotype, it's going to be a blend of the two traits. So basically, what this means, just like they said in the video, is that some traits are not controlled by dominant and recessive alleles. They blend together. So some examples of that might be snapdragons, like we mentioned there, or like the amoeba sisters mentioned. Another example could be, for instance, like a gray dog. That's an example where if a black dog and a white dog had puppies and the puppies came out gray, that would be an example of incomplete dominance. Some other examples, there's another flower called the 4 o'clocks. In addition to the snapdragons, they have either a homozygous red or a homozygous white. The heterozygous will always yield a pink flower, just like you guys can see right up here, pink flower in the heterozygous. So neither allele is dominant. Neither allele is going to take over and be expressed. They're going to work together and blend together. So if we have a homozygous red flower, so capital R, because it tells us in the problem, and we cross it with a homozygous white flower. Now, you can use lowercase r's or you can use a whole nother letter to help you remember this. Um, so our homozygous white flower, it tells us here is W, capital W, capital W. When we do that cross, as always, we bring our top traits down. So this is just like the Punnett squares that we were doing previously. And then we bring our side traits across. Now, as far as genotype goes, we've got 1, 2, 3, 4 boxes, and all 4 boxes are identical, capital R, capital W. That means our phenotype or our physical trait is going to be the same. It's only going to be one possibility. And in this case, because it is the incomplete dominance, it's going to be pink. All of the offspring flower will be pink. So you can see here, this is what our Punnett square looks like, written out a little bit more clearly, and our answers, capital R, capital W for our genotype, because it is the heterozygote. Remember, hetero means different, so it's the mixed one. And now let's see what the Amoeba Sisters tell us about codominance. So incomplete dominance is a blend. Codominance is going to be a little bit different.
Incomplete dominance is different from codominance. Codominance, like a co-worker, that prefix co should make you think together. They work together. The alleles, that is. For that reason, we like to use different letters entirely to represent the alleles. There is a codominance involving color in some types of chickens. Take a look at this Punnett square. If you cross a black chicken, represented here by BB, and a white chicken, represented here by WW, all the offspring here are BW. BW chickens are both black and white. They're speckled. See, both traits show up. This is the essence of codominance. And you know, what's more awesome than a speckled chicken? Well, except for those silky bantam chickens. By the way, you will find that many people use different formatting for incomplete dominance and codominance. Some people like to use different letters entirely for incomplete dominance, and some people don't. Some people also prefer to do exponents with codominance, and some people don't. Formatting aside, when you're solving these Punnett squares, since we find this really does vary from classroom to classroom, the real concept is that in incomplete dominance, one allele is not completely dominant over the other. So you see this almost in-between phenotype. In codominance, both alleles are expressed. It's the application that matters here.
All right. So, like they said, in incomplete dominance, that offspring that is a heterozygote is going to be a blend. In codominance, instead of blending, both traits are shown in the offspring. So, like in our example of our cows here, we can have cows with white fur, we can have cows with red fur, or we can have what we call a roan cow. This one down here at the bottom, that is one that has both red and white hairs spread throughout its body to give it these patches. So we're not seeing a mix. We're not seeing a pink cow here. We're seeing basically patches of each of these traits showing. Another example of codominance is blood typing. And I know that in class we've had a couple of people mention this at different points with different conversations. So basically, codominance has a couple of different factors, or codominance and blood typing has a couple of different factors. There's actually 3 genes that code for blood typing. There's capital A, there's capital B, and then there's O. So O is recessive to everything else. So you'll notice here in our format, we've got capital A, lowercase o, capital B, lowercase o. So type O blood is going to be lowercase o, lowercase o. O is recessive to everything. A and B are both dominant. They're co-dominant with each other, which is why you can get type AB blood. Both are dominant over O. So if you have AO, like my blood type is AO, if you have AO, that means the A is what is dominant and the A is what is expressed. So we just mentioned the genotype a person needs to have in order to get type O blood is going to be lowercase o, because it is recessive. Okay, so if we have a male that has type A blood and they produce offspring with a child that has type B blood, our question here is, could they produce a child with type O blood? Here's the thing. We don't know for sure if dad is AA or if dad is AO. Same thing with mom. We know she's type B, but she could be BB, both of the dominants, or she could be BO. So if both parents are heterozygous, then yes, they could have a child that has type O blood. And here's an example of how. So if dad is type A and mom is type B, but dad is AO and mom is BO, then they do have a 25% chance every time they have a child of having a child that has type O blood. All right, guys, that is incomplete dominance and codominance. So remember, just a wrap up. Incomplete dominance is when we have the two traits, neither one is fully dominant over the other one, so they blend together. They make some sort of an intermediate phenotype. Remember, phenotype is those physical traits. With codominance, they both are equally dominant, so they basically are sharing the spotlight. So, like a roan cow, speckled chickens, a dog with spots on it, those would be examples of codominance. Hopefully that made sense. And I will see you guys soon for our next lesson. We're going to learn about sex linked traits, we're going to learn about pedigrees, and we're going to learn about karyotypes coming up soon. Bye, guys.